It's a great honor for me to introduce this year's recipient of the Boyer Postdoctoral Research Award. Just to tell you about the award, Boyer was a graduate student in this department many, many years ago with Henry Lardy. And then he went to UCLA, where he did groundbreaking research on oxidative phosphorylation, for which he won the Nobel Prize. And then he was generous enough when he got the Nobel Prize to share his prize money with our department and to endow this award. So Chris is receiving Nobel Prize money. How about that? And I'm very proud of Chris, and also of the fact that he's the second awardee from my lab. A number of years ago, we had another postdoc in the lab, Malcolm Kebede, a very brilliant scholar from Ethiopia, who won the award. And she is now a tenured professor at the best university in Australia, at the University of Sydney. So that's no pressure on Chris. So Chris did his undergraduate work at Georgia Institute of Technology, where he studied bioengineering. And then from there, he went to Vanderbilt, where he worked as a technician in a lipoprotein lab and did some excellent work there, and was an author on quite a few publications in the area of lipid metabolism. And then he went to graduate school at Washington University, where he worked on ion channels related to pancreatic beta cell function. When he joined my lab, he was very interested in learning genetics and how we apply genetics to study problems related to diabetes. And he was a quick study. He really did extremely good work, and really is teaching me more about genetics now than I ever taught him. But Chris has developed a real interest in artificial intelligence, and has come up with some brilliant ideas on how to apply that to improve the way everybody does genetics. So that's of relevance to people who do human genetics or model organism genetics. And so he has evolved with his interest in artificial intelligence. And it's hard to recognize him anymore, actually. But I think that it's going to be fascinating to see where that takes him. I think it's going to take him into a very brilliant career future. And so today he's going to tell you about this trajectory, and I think you'll really appreciate how amazing he is.
Thank you, Alan, for that wonderful introduction. And first off, I'd like to say that I'm very excited to receive this award and this recognition, but I'm also very humbled because there are many of you out there who are just as deserving of recognition for your various accomplishments. And I'm really humbled to be a part of this group. And a lot of what you're going to see, and there are so many people I could not fit on this slide, a lot of what you're going to see was made possible by the open and collaborative environment of this department and the university as a whole, without which these projects could not have been completed. And there's so many people to thank. I will give us a couple of shout outs to people who were directly involved with a couple of the projects as I go. But remember, there are so many who were involved in this, and none of this would have been possible without being here. And I'm very privileged to be a part of this community. And so I'm going to break down the trajectory of my time here into three little vignettes that are how I got interested in the role of genetics as it relates to islet biology. First, through a specific project looking at a gene knockout in islets and how to link the loss of this transcription factor to the genes it regulates and the phenotype that we observed. I was interested from that in how we could leverage genetic diversity among the different mouse lines to understand more about islet biology and identify new targets of interest. And then in the last part of it, I'm going to get to what Alan alluded to, which is how we can use some more of these sophisticated methods and leverage these huge data sets that continually get generated by ours and other labs in order to understand how the different components that regulate islet biology interact with one another, which is an area that is central and is of growing interest. Now, my overall interest is in metabolic regulation, and my focus has been on these organs called the islets of Langerhans. Sorry. Maybe this will work better. These are small little clusters of cells in your pancreas that secrete hormones important to regulate nutrients like glucose. And in particular, I'm going to focus on insulin, because if a person's demand for insulin, such as increases, such as when they are obese, but it isn't matched by the production of insulin from pancreatic beta cells, they develop diabetes. And this is a huge worldwide problem. And it's a problem for the research field because islet functional capacity is known to be largely regulated by genetic factors. But the mouse lines that are used to study islet function and diabetes are relatively highly inbred, and they lack a lot of the genetic diversity. So they're missing a lot of the picture on how islets work that is of relevance for human populations. And our lab tries to get around this by using two main populations of mice. So there's a set of 8 inbred mouse strains, including 3 highly inbred traditional lab strains that people have commonly used for these sorts of studies. 2 disease strains, the NOD, which is an autoimmune diabetes model, and the NZO, which is an obesity-driven decompensation diabetes model. And then these 3 strains here, which we call the wild-derived strains, because they were recently developed, they are inbred, but they have been inbred for far fewer generations than the other lines, and they are in no way related to one another or to those other lines. So they represent a pool of added genetic diversity. And collectively, across all of these lines, we get a high diversity in metabolic responses, and we have a high diversity in their genomes. And by crossing these, by outcrossing them and shuffling their genomes around for multiple generations, we've generated stocks of mice, or I should say others have generated these stocks of mice, and we use these stocks of mice, called the diversity outbred stock. And this gives us sufficient power and environmental control to ask really detailed questions about how we can associate regions of the genome with particular traits we're interested in. And we call these quantitative trait loci, or QTL. And these peaks allow us to identify new genes that may be candidate regulators of islet biology and metabolism. And so a previous study that was completed before I joined the lab took 500 of these diversity outbred mice, fed them this high-fat, high-sucrose Western-style diet to stress them. And from islets that were isolated in these mice, some of them were subjected to functional characterization, looking at insulin secretion in response to multiple secretagogues. Some of them had their transcriptome sequenced, and some of them had their proteome determined. And from all of this, we can integrate these data to identify new regions with candidate regulators that may be important for islet biology. And one of these that is the subject of my first vignette is this transcription factor called ZFP148, or ZNF148 in humans. And so this is a transcription factor that's associated with the development of certain organs and in certain cancers. We think that there was strong reason to think that it had a role in metabolism because it's known to interact with a number of transcription factors that themselves have independently identified roles in metabolism and islet function. And in other tissues than islets, it regulates components of hormone signaling pathways that are relevant to islet hormones, like insulin receptor signaling. And if you look at the transcriptional abundance of this gene across the different tissues that have been surveyed by different groups, including by this huge set that was surveyed by the Tabula Muris Consortium, those tissue cell types that most strongly express this gene are in islet endocrine cells, including the insulin-secreting beta cells. And so the final piece that this would be important for diabetes risk, the final clue rather, is that the orthologous gene, ZNF148, has single nucleotide polymorphisms in human genome-wide association studies that confer type 2 diabetes risk sitting right next to it. And we know from studies that others have done looking at physical contact between different regions of the genome in human islets, that these SNPs are in areas that make physical contact with the promoter of this gene. And this suggests that they confer diabetes risk by regulating the expression and abundance of this transcription factor. And we study this using a system where we can selectively knock out this transcription factor only in beta cells, because we cross a mouse that expresses this Cre recombinase under the insulin promoter, which turns on as soon as the islets start making insulin, to a mouse where ZFP148 is flanked by these LOXP regions. And for mice that express both of these traits, the ZFP148 gene is spliced out. And so we get a tissue selective knockout, which we can characterize. And so looking at the glucose responses of these animals, and so here, this is time following a glucose, oral glucose challenge on the x-axis and then plasma glucose on the y-axis. I hope you can appreciate that the controls in white peak with a higher glucose and take much longer to return to their basal glucose than the ZFP148 beta knockouts in red. And this is true irrespective of sex, And it's also true even when we challenge them with this high-fat, high-sucrose diet. And so this represents a dramatic improvement in glucose tolerance. And we know from the fact that other phenotypes like body weight, fasting insulin, and other traits are not different between these animals, that this is a very islet-centric phenotype. And that makes sense because we're knocking it out in beta cells. And so if we take the islets out of the mouse and we subject them to a flowing solution of different nutrients, we can look at their insulin secretion. So this is time on the x-axis. This is insulin as a percent of total secreted on the y. And you can see that following a transition to a stimulatory concentration of glucose, the insulin secretion in the knockout islets shown in red is much greater than the insulin secretion in the control islet shown in white. And this is true also if we do a static-based well assay, where we just put the islets into a solution and leave them for a period of time before measuring insulin. Now, this raises the point, though, that insulin secretion is pulsatile. It oscillates in the body. These pulses of insulin secretion are important for how insulin signals in the periphery. So they're important for metabolic regulation. And one of the hints that diabetes is developing is that these become dysregulated as the course of that disease progresses. And so we know that a number of the mechanisms that control insulin secretion, such as the metabolism of nutrients, and the oxygen consumption in that process is shown here, as well as the membrane depolarization that's triggered when the ATP that those nutrients produce closes these KATP channels, an ion channel in the membrane that's sensitive to ATP, as well as the influx of calcium that results when the membrane depolarizes, and which is itself the most proximal trigger for insulin secretion. All of these pieces oscillate. And their oscillations are characteristic of different pathways that regulate insulin secretion. Several groups have elegantly shown, including the Marin's group here at UW-Madison, that if you understand the parameters that describe these calcium waveforms, you can infer very detailed mechanistic data about what's going on under the hood in the beta cells of the islet. And so to look at this in the ZFP148 knockout islets, I used a trick where I can take islets from an animal of one genotype, label them with this lipid dye called dye R, and then mix them with animals of a different genotype and stain all of them with a calcium sensor dye called fluorescent red. And this lets me image both the knockout and the control islets in the same dish under the same perifusion conditions. So I can look at their responses to the same nutrient stimuli simultaneously. And this is very, very powerful for high throughput studies looking at islet calcium. And so looking at an example trace like this, so time is on the x-axis, and then the calcium is indicated by the fluorescent red ratio is on the y-axis. You can see that for the control in gray, as I ramp the glucose from a non-stimulatory 3 millimolar all the way up to 9 millimolar glucose in steps, that at certain steps of intermediate glucose, these islets start to pulse, their calcium starts to oscillate. And what I hope you can appreciate is that the beta knockout islets begin their oscillations at a much lower concentration of glucose, suggesting that they have altered nutrient flux and different nutrient sensitivity. And this was on chow. This is true also if we put the animals on that high-fat, high-sucrose diet. And what I hope you can appreciate here is that where the control islets now begin oscillating at a higher concentration of glucose, suggesting that their beta cells have been stressed by this diet, the knockout islets continue to oscillate at these lower concentrations, suggesting they are resistant to the effect of the diet on beta cells. And so looking at some of the parameters that describe these calcium waves, I'm going to show you here one of them, which we call the plateau fraction or duty cycle. What I hope you can appreciate is that this defines basically the fraction of time for each pulse that the islet is in the active and secreting state. And if you look at it, across all of the different glucose concentrations shown here on the x-axis for the summary graph, the duty cycle is elevated in the knockouts relative to the controls at all of those different conditions. And this is consistent with a much higher flux of nutrients through the different nutrient uptake and metabolism pathways. So what mediates this shift in flux and this shift in sensitivity? So to get at this, we did RNA sequencing on islets from the knockouts and the controls. And we found about 650 differentially expressed genes, of which about 2/3 of them are increased in the knockout versus the control. This is consistent with what's known about the role of this transcription factor as a repressor of gene expression in most other tissues. But we wanted to get at which of these 650 genes might be responsible. And one way that I did this was to look for the most direct targets of ZFP148. Now, there is no ChIP-seq data set in islets in either humans or mice, but we were able to leverage the fact that through ONCODE and other studies, there was ZNF148 binding peaks were known for humans in these other tissues. And so I looked for orthologs of the differentially expressed proteins, such as PSAT1 and GLIS2, and I asked whether peaks had been found in other tissues for ZNF148 binding for these orthologs, either right at their start on their promoters, or even if they were far away, whether they were in regions that made physical contact with the promoters of these genes as determined through this promoter capture Hi-C data that was taken in human islets. And of all of the genes that we looked at, about 200 had met these criteria as our putative direct targets. And it was interesting because the gene enrichment for ontology enrichment for this subset of the genes included terms like glycolysis activation in cancer and this metabolic reprogramming term that is also associated with cancer. And the genes in this category are associated with shifts in nutrient utilization and sensitivity. And so it was really interesting to us whether these would result in a difference in protein abundance. And so with the help of the Attie Lab, we did proteomics on the knockout and the control islets. And you can see the proteins that are upregulated here and or increased here and decreased here in the knockout versus the control. And of the 6,000 that were measured, only about 200 were differentially abundant. And for those who use RNA as your go-to for pathway enrichment analysis, I do want to caution you that none of the most changed genes in our RNA-seq data set were the extreme genes in our differential abundant protein set. So it's very important to not just go by what's the biggest change for your RNA. You really have to validate that. But what was very interesting in this is that the top pathways that were enriched in our differentially abundant protein list matched the list of metabolic reprogramming terms that were seen for the putative direct targets of ZNF148. And they included these glycolysis activation terms and this glutamine reprogramming in cancer. And so taking all of the differentially abundant proteins, I asked whether any of these fit into pathways that could potentially alter nutrient handling in beta cells. And so one area that's of critical interest in many labs is looking at this cycle called the phosphoenolpyruvate or PEP cycle. And so PEP that's produced from glycolysis or other sources is converted to pyruvate by pyruvate kinase M2. This produces the ATP that closes these ion channels called KATP channels and triggers insulin secretion. So it's of great interest to a number of people. But there are a lot of pathways that feed into it. And so pyruvate can be converted to oxaloacetate instead of going through the TCA cycle, and then that can be converted to PEP. Or you can get carbon entry through these other sources like succinyl-CoA and alpha-ketoglutarate that can then feed into oxaloacetate and then to PEP. And so there are a number of different ways that the proteins that were increased, and I'm showing these as highlighted in blue, that are increased in the knockout versus the controls, could feed into this. And they include the production of an activator of this enzyme, serine. They include the increased flux into the cell through these transporters that are elevated in the knockout. And there are enzymes involved in glutamine metabolism, including both in the conversion of glutamine to glutamate and then glutamate's entry as alpha-ketoglutarate that would mediate entry of carbons into this branch of the TCA cycle and then thereby provide it as a source for PEP production. And so I wanted to see whether this could be true. And I looked at this with imaging and secretion studies by using low glucose to minimize the amount of PEP that's coming from glycolysis, and then adding the amino acids as the nutrient source. And so, So in this study, all of this is at low glucose. I gave a mix of amino acids where the number in this amino acid ramp reflects its relative concentration versus its physiologic concentration for each amino acid. So physiologic, twice physiologic, 3 times physiologic, et cetera. And you can see that the beta knockout islets, shown in red, have an increase in their calcium at low glucose in response to this amino acid ramp that's elevated above their control counterparts at all of the concentrations we tested. And this is also something that we can see if we give a pyruvate kinase activator to to maximally open the door, metaphorically speaking, and see whether the capacity to flux carbons through this pathway is just elevated in general in the knockout. And so when I do that and repeat the ramp, you can see that they're still elevated in many of the concentrations of amino acids, and the calcium is elevated in the knockouts versus the controls. Now, I'm showing it just for females on chow here, but it's also true in males and when we treat them with a high-fat, high-sucrose diet. So this suggests that the amino acid pathways overall are changed, but the glutamine enrichment term suggested another experiment as well. And so glutamine is converted to glutamate and converted from that to alpha-ketoglutarate, but the enzyme that does the final conversion, glutamate dehydrogenase, is normally inactive without a factor to activate it that's produced from leucine metabolism. And so we can mimic that intermediate metabolite with a compound called BCH. And so we can give glutamine and low glucose. And when this is present on its own, it doesn't stimulate secretion. But as I ramped up the concentration of this activator compound, BCH, you can see that the insulin secretion was elevated in the knockout versus the control, suggesting that its capacity to flux glutamine carbons into the TCA cycle is also elevated. And so the summary of this project is basically taking an unknown gene candidate, this transcription factor, identifying the pathways it regulates and how it pushes carbon through these cycles to alter nutrient sensitivity and insulin secretion. And this is a specific example of an understudied target, but I wanted to really understand whether we could identify novel proteins and targets through other methods by leveraging the genetic diversity of these different mouse strains. And so this moves me into the second topic of this talk, which is a project that I worked on in conjunction with Lauren Clark, a grad student in the lab. And so these 8 mouse strains have a lot of genetic diversity, and it's possible to do studies in them that we can't do in the diversity outbred mice, just for practical reasons. And so one of the previous studies that Kelly Mytok in our lab did showed that the insulin secretion of these animals' islets in response to these different secretagogue conditions is widely varied between the strains and sexes. Now, this is a static secretion measurement, and we know that understanding the oscillatory behavior of these islets is very critically important for establishing the different mechanisms that may be at play in the diversity of those responses. And so we asked whether the calcium oscillations would similarly be variable among the islets from these different founder strains. And so we took mice that were from these strains and both sexes and fed them this high-fat, high-sucrose diet, and then isolated their islets, stained them with calcium dye, fluoro-red, and then imaged them, looking for changes in oscillatory response to 8 millimolar glucose, which was our interrogator of beta cell function, 8 millimolar glucose with amino acids added, which interrogates this alpha to beta cell communication in the islet, and then looking for whether the islets respond differently to gut hormones by adding this glucose insulinotropic polypeptide hormone. And then we get a measure of basal calcium by washing all of that out in low glucose. And so when we look at this in example traces from males in all of these lines, you can see that there's a wide variation in the types of responses in calcium to these different nutrients secretagogues across the different strains. And they tend to cluster in certain ways. So, so the inbred lines, the traditional ones that a lot of labs use, tend to be very similar. They tend to have much slower oscillations that reflect a lot more metabolic mechanisms being at play. And they tend to increase their activity with amino acids, but return to these oscillatory behaviors when we add the gut hormone, GIP. Whereas the wild-derived strains that are very different from the other inbred strains and from one another, nonetheless appear very similar in their calcium responses. They tend to be dominated by these super fast oscillations that invoke more ionic mechanisms. They tend to max out with their amino acid exposure, but they don't return to oscillatory behavior in response to the GIP treatment. And so the disease strains, the NOD autoimmune and the NZO obese-driven diabetes models also have their own unique signatures that reflect different aspects of their disease pathology. And so we wanted to ask whether components that describe these waves, and I'm showing a handful of them here. I'm not going to go into detail about this for time, but we wanted to ask whether these were reflective of different mechanisms that might be happening in these different islets. And so one of the ways that we do that, And we know that this can happen because, for example, when you pharmacologically inhibit that gatekeeper ion channel KATP, it changes the time between pulses, but doesn't really change their overall shape very much. Whereas if you close it by increasing nutrient production and ATP synthesis, then the shapes change in multiple ways. And these sorts of changes are ways that we can interrogate which pathways may be involved. And some of the parameters that describe these calcium waveforms, if we look at their variation by sex and strain and correlate that to the variation by sex and strain for insulin secretion, for example, some of them, like the silent duration and the amino acid condition, very strongly correlate with insulin secretion across multiple different secretagogues. Whereas others, like this first frequency component in the 8 millimolar glucose condition, are very anti-correlated. And this suggests that they are targeting different pathways to alter insulin secretion. And they also correlate differently to different in vivo traits, like plasma insulin, as I'm showing here. And the patterns of this can be quite informative because, sorry, I jumped ahead there. Because we also had measurements from the prior study of protein abundance in the islets from these strains and sexes. And you can see that the protein abundance varies quite widely among the different mice. And so if we ask whether the strain and sex variation in those proteins correlates to the sex and strain variation in the calcium oscillations, you can see here that individual proteins are shown by each of these little columns, and they cluster for ones that are very highly correlated to specific traits, and they have blocks that are associated with traits of one kind, like the silent duration there. And so this suggests that each of these calcium oscillation parameters is associated with a specific subset of proteins that regulate a key pathway involved in the islet response. And if we look at any one of them, and for example, take the tails, the most correlated or anti-correlated proteins to that given parameter, and run them through pathway enrichment, they enrich for their own subsets of key pathways. And some of these, like this insulin biosynthesis pathway for this set, are relevant for islet biology. And so we can leverage this to identify novel pathways that may be important for regulating islet calcium responses. Now, we need to know which of these are relevant in humans, and so one way to do this is to take those correlation tails, look for the orthologs of the proteins in those tails that exist in humans, and ask whether any of those orthologs have single nucleotide polymorphisms for glycemia-related traits in humans. And so we can see whether they do above the gene body itself, like they do for this COB-LL1. You can see the different glycemia-related trait SNPs there. And sometimes also they occurred for orthologs where the SNPs were not over the gene, but made physical contact with the gene's promoter in promoter capture high C data. And this took us from a list of over 3,000 proteins that were correlated to one or more of our calcium traits down to just under 650 that met all of our criteria, including having the glycemia-related SNPs. And then through a light triage, there are about 480 that are relatively understudied for their roles in islet biology. So this is a large list of targets, but the ones that were known for having a role in islet biology did emerge among the 650, including those involved in metabolism, ion channels, and calcium influx. And this suggests that this pipeline is robust and a good method for condensing this large list of proteins down to the ones that may be most relevant for islet biology. Now, in conjunction with Brian Yandel, we've created a resource that displays some of this data. So if you have a protein you're interested in and you want to see what it's doing in islets, you can go here and plug it in and see how it varies among the strains and sexes and whether it correlates to one or more of the calcium traits. But we also wanted to use this to discover new targets. And I'm going to tell you stories about two targets that we went after based on this study. And so one of them is transketolase, or TKT, and the other is this dipeptidase 8. And based on our correlation data, we predicted for both of these proteins that if we knock down that protein, it would increase insulin secretion. And that's important for you to remember. So looking to the first one, we wanted to see whether this would change insulin secretion. So the way that I do this is to transfect islets with viruses that express shRNAs to knock down the gene. And so if I do this with a relatively low dose of virus, I get a modest knockdown of the gene, but I still get a robust increase in insulin secretion. And I see this in multiple strains of mice, the B6 and the 129. I see this in both sexes. These are males and these are females, and this is on different diets as well. So this is a robust increase in insulin secretion in response to the knockdown of this gene. And in human islets that I knock down the human ortholog with these conjugated siRNAs, I still see an increase in insulin secretion in these islets. So this suggests that it is not only a robust regulator of insulin secretion, but it may be relevant for human biology. Now, I'm going to move into the final part of this by talking about the second gene that we went after, which is this DPP8. And so we don't have a gene knockout of this, and we don't have a virus knocking it down, but there is a selective drug that was developed because it's implicated in certain cancers and autoimmune disorders. And so what we found, contrary to what we had expected, is that when we treat with this inhibitor that blocks DPP8 activity, whether it's insulin response to glucose or amino acids or GIP, it seems to be dramatically suppressed in islets that were treated with the inhibitor for this protein. And this is also true in the static secretion assays that we do. And so the question arose, and I was really interested to explore whether there was a way that we could go beyond just the simple correlation between a gene and a trait to identify correctly the degree and direction of effect these candidates might have in regulating islet function. And so this is important for the field at large because while the disease of diabetes is highly heritable and islet function is largely driven by genetics, the single nucleotide polymorphisms, like the ones that I showed you previously for ZNF148, those account for only a small fraction of what we know is a highly heritable trait. And this means that there are other factors at play. And this makes sense because proteins have binding partners, they're post-translationally modified, they change their localization in the cell. And the way that we analyze these data for these single nucleotide analyses and the kinds of analyses we do with the DO doesn't really track well the gene by gene interactions or the gene environment interactions that we know play critical roles in diabetes development. And so I wanted to take some of the vast amount of data that we've generated, the insulin secretion, transcriptomic, and proteomic data, and see if there was a way to reduce the complexity of that data that still preserved the potential interactions between some of the key variables and might hint at which proteins were conditionally dependent on other proteins for their effects on islet function. And the way that I did this was to use machine learning algorithms. Now, for those who haven't used them, and I'm not an expert, and I'll qualify this that way, these aren't magic. They're not a black box. Most of them are ways of subcategorizing and independently analyzing parts of your data set. And they're useful because they can preserve some of these interactions, but limit the number of variables that you have to look at. So instead of thousands and thousands of proteins, you have a small handful, and you know how they connect together. And so this is powerful because we can run multiple types of data through the models that we generate, not just the data we use to generate the model. So we can see whether our conclusions apply in data sets that others have generated. And because we can do this with data that was generated in the diversity outbred mice, we can also ask genetic questions for the proteins that we're looking at. And so the way that I do this is that I take islets and the insulin secretion data and proteomic data from about 370 of these DO mice. I split the data where about 70% of the mice are used to train the model and build these equations that determine insulin secretion based on protein abundance. And then I use the remainder for validating the assumptions that that model was generated. And so if I do this, this is showing the training set validation for a model that predicts plasma insulin and its comparison to what the actual plasma insulin was. You can see that it's reasonably well correlated. It's a constant of about 0.7. And this is similar to what others have shown for predicting traits in highly variable populations, including this study that was done in the DO looking at exercise tolerance. It's getting about the same order of correlation for its testing set. So this was encouraging. We also had human data, and I wanted to see whether this model modeling approach would work for human islet data. So Jim Johnson's group collected proteomic data and secretion data for about 140 islet donors. And our lab, in conjunction with Lloyd Smith's lab, had a handful of donors where we collected proteins and its secretion in response to short-term low and high glucose treatment or an intermediate term in these different secretion conditions. And so I wanted to see if we could build models with this data. And so if I take the Johnson lab data and I feed all of the protein abundances into it to build the model, I get a correlation that's not quite as good as I had seen in the DO, but is still reasonable for such a diverse population of human data. And if I feed the secretion data or the proteomic data that we measured in the 9 donors in conjunction with the Smith lab for these 9 type 2 diabetic and normal donors, we don't have an exact match for the glucose secretion condition the way that the Johnson lab had it, but secretion in our stimulatory glucose is predicted reasonably well. Now, it's a small number of donors. You have to take it with a grain of salt for that reason. But it suggests that we can take the data sets that were generated independently and use them in models that were, or use them in models that we generate with other data. And so looking at that model again that was generated in the DO mice that predicts plasma insulin, I can take the data that we had in a separate study of the founder mice and plug the islet protein abundances into it and predict the plasma insulin. And you can see that it correlates very well with the actual plasma insulin that was measured in those founders. Now, of course, this comes with the caveat that both of these studies were done in our lab. And so the question is, would it work for a study that was done far away by other sets of hands? And so fortunately, Malcolm Kabede in Australia did a parallel study in which they measured plasma insulin and protein abundance in islets from these different strains of mice. And if I plug their data into it, the predicted plasma insulin correlates extremely well with the actual plasma insulin. And what's even more encouraging is that the phenotypes cluster, so both the NZO mice shown in this red circle and the OB and DB mice here are models of weight or obesity-driven islet dysfunction and diabetes. And the fact that the phenotypes cluster in the same regions of the predicted model suggests that it's keying off of pathways that are relevant to islet biology rather than some weird noise or batch effect. And we have other clues that these models may have relevant information for identifying new islet targets. And so if we look at a list of proteins, the top most influential proteins for the predicted outcome, and this is showing their importance to the model here on the x-axis, and this is the top 15 or so of proteins in this model that was predicting plasma insulin. You can see that some of them are known, and I've highlighted those with these red diamonds, and others have GWAS SNPs for glycemia-related traits, and I'm showing those in blue. And they enrich for pathways that may be relevant for islet biology, so for membrane trafficking and for cell types that are cell types found in the pancreas. And so what I want to do eventually is to be able to feed data that we get from humans where we have far fewer ends, there are fewer donors, into models that we generate in mice where we have a lot greater power and environmental control. And so what I'm working on now is figuring out ways to harmonize the data between them so that this generates equivalent accuracy. Because right now, when we feed the Jim Johnson's lab's islet data into our models generated in the DO mice, the correlation isn't so great. And we don't have the exact same conditions. That's a caveat that goes with this. But I still want to figure out ways to improve this and to get the data more harmonized so that we can ask questions about what drives islet biology in humans using the DO mice as our tool. And there is an additional method that we can use with the DO mice, and the reason that I want to use them as a model system, which is that we can look at the feature significance either across the whole data set for a model, or we can look at it on an individual mouse by mouse basis. And so this is the prediction, the change in the predicted insulin. And you can see that as you start from nothing, you can add, each of these proteins predicts a certain addition or subtraction to that prediction. And adding them all up gets you to what the final prediction is. And for some mice, like this one, the profile of proteins from most significant to least significant and their relative influences on the actual prediction are similar to the overall population, where others like this mouse, are very, very different. The most influential protein is not this secretogranin 3, and secretogranin 3's effect is actually the opposite in this mouse of what it is in this mouse. And this suggests that these may be influential, these metrics may allow us to identify what works on an individual mouse-by-mouse basis. And since, and then I'm going to return to the example that I started this whole segment off in, which is DPP8. So we have ways to extract the feature significance for any of these proteins. So if there's a model where DPP8 appears, would it accurately predict the relationship that we observed when we experimentally manipulated DPP8 activity? And so for this model that predicts secretion in glucose and GLP-1, I can take the importance of DPP8, that Shapley value is what we call it, its influence on predicted secretion, and correlate that to its abundance. And that correlation in this case is positive. So the more of it you have, the more positively it influences your predicted secretion. So the more secretion you're predicted to have. And this is consistent with what we directionally observed, whereas the correlation alone predicted the opposite. And this suggests that some other proteins are interacting with DPP8 to influence the impact it has on islet function. And so this is true for multiple different proteins in these models. Some of them, like this one, ADH1, have influence patterns across the different mice, and each of these columns is an individual mouse that resemble their abundance values, suggesting that what controls how abundant they are is what's controlling their influence on secretion. Whereas others in the same model, like secretogranin 3, have patterns that in no way match their abundance patterns. And this suggests that what controls how abundant secretogranin 3 is not necessarily what controls when secretogranin 3 is important for islet biology. And so we can ask questions with the diversity outbred mice using the variation in these SHAP values as a meta trait. And we can look for statistical associations between regions of the genome, and I'm showing the different markers we look at across the chromosomes here on the x-axis, and their relative statistical association to the trait that we're interested in. For example, the value for the Sec24. And where they are above our significance threshold, we can look for candidate drivers at that position, genes that may be important in influencing the association between that marker and the phenotype that we're observing. And so because we look at these in the diversity outbred mice, these haplotypes at each of these different positions can be traced back to the probability that they were inherited from one or more of the parent strains. And so for a trait like this, for Sec24, the mouse that had the inherited its allele from the PWK line has a very low value for the SHAP value for Sec24, whereas those that come from the 129 have a very high value. And these sorts of allele effect patterns are important because genes that drive the phenotypes we're interested in and at the positions we're interested in often share that kind of pattern. And for example, SAT2 at this position looks very similar in its allele effect distribution to the Sec24D value for its influence in the model that it came from. And we can use other tricks, like including the expression of these genes as covariates when we do our statistical analysis looking at these different markers. Or we can use these other modeling softwares that other labs have generated, like this Bayesian prediction system that Gary Churchill's group created, to identify which genes under these peaks may be the drivers of that phenotype. And so these together, altogether, these models and the ways that we can manipulate the feature significance values for them, how important the proteins are predicted to be, allow us to identify conditional dependence between different proteins, and they allow us to identify regions of the genome that may be important for how those proteins are influencing insulin secretion, and not just how abundant they are. And so this is all preliminary. There is still a lot of work to be done in this. But I want to figure out for my next steps if there's a way to harmonize the human data so that the mouse data and the human data predict equally well. I want to, as part of that, see if I can incorporate RNA in some fashion into the modeling, because the read depth for RNA is greater. There are a lot more RNA data sets in humans than there are proteomic data sets. So this may help me build new ways to harmonize the two data sets or the two data types. And finally, because I'm relatively new to ML and because I'm not a statistician by training, I really want to go through and vet some of these. I picked the modeling type that I used for the data that I presented here based on the fact that it had been used previously in other DO cohorts for different traits. But that doesn't necessarily mean that it is the right model for the data types that I'm attempting to analyze, like proteomics and insulin secretion. And finally, I want to know that I'm using the best method to extract the weights of these different proteins in the models, if I'm getting the most information that I can out of them. And so all of this is what I'm currently working on, and it's the direction I'm taking for my training and transition award. And it's kind of the culmination of everything that I've been doing here, defining these different interacting networks and trying to identify and leverage our data so that we can determine these new targets regulating islet biology and metabolism. And with that, I will take questions. Thank you. Anyone? Go ahead.
Thanks. That was a great talk, Chris. I'm curious about like your machine learning models because, you know, you mentioned that in some instances between the cell types, different genes will have kind of different levels of basically like their expression and whether that correlates to a particular phenotype. You know, one reason that could be the case is if you were too aggressive with your dimension reductionality in the machine learning model, and you've essentially removed factors that are key to describing those differences accurately. Have you tried running models even of the same type with different stringency, or like how are you approaching kind of when you feel like the model has been sufficiently trained but isn't overly trained so it won't be generalizable?
So what I've generally done has been kind of like there hasn't been a rigorous system to it yet. I'm really new to this. So I have played with some of the variables to try to get the models to predict more accurately. A lot of what I've been going by is actually the histogram. So of the different predictive accuracies, when I go through and I train the models on randomization of those data sets. And for the most part, I've been sort of playing that by ear. I haven't had a really rigorous kind of testing regimen for it. But that's one of the things that I want to do what I mean when I say that I vet the models is go through and really rigorously establish whether those parameters are correct. Because I said a lot of this by what was published. And that has proven interesting, but it doesn't necessarily mean that it's optimized well. So thank you. It was a great question.
Hi, so I had a question regarding the ZFP part of your work. I was curious to see that like the amino acid sensitivity was different compared to non-knockout. I'm wondering on whether that amino acid sensitivity was collective or whether you were able to discern more various types of amino acid metabolism, especially going back to how you were talking about glutamate dehydrogenase being affected, of whether you were able to find any more, and maybe even discerning that further, whether there was like differences in, say, non-essential versus essential amino acids, since that's diet correlated.
So we did only one subset of the amino acids, which was looking at glutamine metabolism. That was suggested by that specific pathway enrichment. So overall amino acids are different. Glutamine metabolism specifically is also different. One of the series of experiments that I've wanted to do, and we were getting started, was to look at serine metabolism as well, because we know the serine biosynthesis pathway is affected. And I didn't show it here, but there are other enzymes important in serine and 1-carbon metabolism that are also different. So it's certainly not just glutamine or all of the 12 that are combined, but we're still figuring out ways to parse which ones are the most significant. So I hope that answers your question. Yeah. Go ahead.
So earlier in your talk, I understood you to say that there was a very poor correspondence between mRNA levels and analyzed by RNA-seq and protein levels determined by proteomics. Is that right?
That's what we've observed.
But now you're talking about, putting RNA levels into the model.
Mhm.
Is that going to make things better, or do you have any idea what the basis is for that
So
very
I've
poor
tried.
correspondence?
I don't know what the reason is for the poor correspondence overall. We know from islet preps, particularly for human islets, people have observed that they're very subject to batch effects. They're isolated at different centers all over the country. The people they come from have been dead for different causes, different times, where the proteins don't seem to be as affected by that. In the long run, if I could work with proteins mainly, I would work with proteins mainly. But this is also something of a practical aspect, which is that there's just a lot more RNA data out there. The other thing that I've noticed going through this, I don't know if I have it as a backup slide in this, hold on just a second, is that when we're trying to feed the different data sets that we did not generate into models that were generated with data sets either that we had in-house or in the case of the Johnson lab data from that data set, the degree of overlap between the number of features that was measured in both data sets is a huge factor in how well they perform. So the DO data, for example, only has about 4,500 proteins that are in common with the Johnson lab human data. So they measured about 8,000. And so if I take a different data set, if I take Malcolm's data set and feed it in there, because they had about 8,000 proteins measured, the correlations that I get are stronger, the fits are better. And that's just because more of those variables are present and don't have missing values. So that is kind of why I'm leaning toward finding some way to incorporate RNA. I do want to find a way to get around the batch effects because I know that those are important, but just having the ability to work with all of that extended data would be very powerful, I think. I don't know if I have that as a backup slide. Oh yeah, and we can make models with the RNA that work in reasonable range too. So that's what this is. I don't have the other one. Okay. So other questions?
Okay, thank you, Chris.
Thank you.